Welcome back. Today we are going to discuss about chromosomes. In this video, we will be describing about the chromatids, centromeres, secondary constriction, telomeres, and obviously types of chromosomes. So let us start the video. You know that the genomes of prokaryotes are contained in single chromosome, which are usually circular DNA molecule, whereas the genome of eukaryotes are composed of multiple chromosomes, each with linear molecular DNA. That is very well known to you. So let us know what chromosome means. Actually, the chromosome has been derived from two words. Chroma means color and zoma means body. But I must tell you that the chromosome is actually not a colored structure inside the cells. Rather, they appear colored when cells or nuclei are stained with certain dyes like acetocarmine or acetoarsine solution. Okay, so normally they are not colored, but they stain when they they appear colored when they are stained with dyes like acetocarmine or acetoarsine. Got it? So chromosomes are compact physical structures which are formed of chromatins on coiling. Got it? So we do say that chromosomes are the physical basis of life. Okay? So what is the history regarding chromosomal discovery? Hofmeister in 1848 first discovered chromosomes in dividing cells. Then your Strasburger in 1875 observed chromosomes during cell division. Got it? So Waldeyer in 1888 coined the term chromosome. So these were the various historical point of view regarding the discovery of chromosome. Now let us come to the morphology of chromosomes. So how it appears externally. As far as your chromosome is concerned, we are generally concerned with the metaphasic chromosome. Why? Actually, during cell division, the chromatin condenses, and by the end of prophase, they become distinct thread-like structures called chromonemata. Singular is chromonema. So during cell division, what happens? The chromatin condenses, and by the end of the prophase, they appear as a distinct thread-like structures called chromonemata. So after that, in metaphase and in the anaphase, the chromosomes become fully condensed and take the shapes of your chromatids. So this cyclic change of size of chromosomes during cell cycle is called chromosomal cycle. Okay. So let us consider about the metaphasic chromosome. A metaphasic chromosome is generally described when we talk about the chromosomal structures because of their distinct structures. These are generally studied for morphological purposes. Got it? So a metaphasic chromosome consists of chromatids, then your centromeres, which are otherwise called as primary constriction, then your secondary constriction and satellite body, then telomeres, then these are the structures as far as morphological structure of chromosome is concerned. So now come to the diagram of your chromosomes, structure of chromosomes. As you can see quite easily that here there is presence of your secondary constriction, then there is presence of your satellite body, there is presence of a secondary secondary constriction and primary constriction. Okay, these are certain structures we will describe in details in the next slide. So first come to your chromatids. Okay, as far as chromatids are concerned, these are the two longitudinal halves of a metaphasic chromosome. Okay, the two chromatids are formed during the synthetic phase of the cell cycle. You know the cell cycle, the G1, S, G2 and the M phase or dividing phase. So actually the two longitudinal halves of the metaphasic chromosome, which are otherwise known as chromatids, these are formed during the S phase of cell cycle. how does that how do the two chromatids are formed? It is because of the replication of a single DNA molecule. You know that each chromosome is made up of a single DNA molecule. Now during the S phase, the replication of DNA occurs. That means a single DNA molecule gives rise to two DNA molecule. A replica of it is formed. So ultimately a single chromosome now is made up of two chromatids, two longitudinal halves of a chromosome. Okay, so two chromatids are actually genetically identical, hence these are called as sister chromatids. Okay, now the two chromatids are joined at specific point called centromere by a protein called as cohesin protein. Okay, and each chromatid is made up of a double-stranded DNA. Okay, so this is how it looks. If you draw the diagram of a chromatid, you can quite easily see that this is the single one, this is the, yes, these are the two longitudinal halves of a chromosome, then this one. Okay, so now as you can see here, this is the centromere, this is the centromere, then this one is the chromatid 1, okay, and this one is the chromatid 2. So we can say that these two chromatids, first chromatid and second chromatid. First chromatid means this entire part, second chromatid means this entire part. So there are presence of two chromatids, and these two chromatids are genetically identical. So they are known as sister chromatids. Got it? Okay, next come to the centromere or kinetochore. Centromere and kinetochore. So what is this centromere? The centromere is a highly constricted region which is otherwise known as primary constriction. Okay, so that constricted means a narrow zone. Okay, it looks constricted. Why? Because in this part the DNA is without histone proteins, nucleosome, okay, and is decondensed. We will discuss about nucleosome in the fine structure of eukaryotic chromosome. Okay, so you remember that the primary constriction or centromere, which is otherwise known as primary constriction, why it is constricted? Because DNA here is without histone proteins and without nucleosome and is highly decondensed. Got it? Then it actually divides the chromosome into two equal or unequal parts, which we call as arms. The parts that we are saying are called as arms. So the centromere is dividing the chromosome into two equal or unequal parts. There is another structure called kinetochore. Okay, so what is a kinetochore? I will show it in the next slide. The kinetochore is a disc-like protein. Okay, disc-like protein which is formed at centromere during prophase. Okay, disc-like protein formed at centromere during prophase. Kinetochores, what is the function of kinetochore? It is the site of attachment of microtubules that give rise to spindle fibers. Got it? So it is the site of attachment of microtubules. So let us see the diagram now. In the next slide, you can see quite easily that this one is the two chromatids. As I had drawn in the previous slide, the two chromatids. This one was a chromatid, then this one is your centromere region, then this one is the second chromatid. Got it? So chromatid 1, then chromatid 2. Okay, as you can see here, this one is chromatid 1, then this one is chromatid 2. Okay, now as compared to this particular part, this part is a constricted part. So constricted part means it is the primary constriction or centromere. Okay, in this primary constriction zone, there is presence of a kinetochore, disc-shaped kinetochore. This yellow part is kinetochore. This one is kinetochore, to which the spindle fibers are attached. You will know in details about the function of spindle fibers during the mitosis phase, during the cell division stage. I will discuss it in details about the spindle fiber. What is the function or what is the role of spindle fibers in mitosis or in during the cell division process. Got it? So in short, I can say that the primary constriction or the centromere is a narrow zone or constricted zone. This one is the constricted zone. Now if we are saying it is constricted zone, why it is constricted? Because here DNA is without histone protein and also nucleosomes are absent. Got it? So, what is the function of this centromere? This centromere is actually dividing the chromosome into two equal or unequal parts, which we call as arms. Okay. Then what is a kinetochore? The kinetochore is a disc-shaped protein which is attached to the centromere during the which is formed at the centromere during the prophase stage. Okay. And to this kinetochore, the spindle fibers are attached. Got it? So this is what the structure of centromere and kinetochore. Now let us come to the next one, that is the secondary constriction and satellite. As far as secondary constriction is concerned, it is the constriction beyond primary constriction. Okay, so beyond the constriction which is present beyond primary constriction is called as the secondary constriction, which is otherwise known as nucleolus organizing region. Okay, why we call it as nucleolus organizing region or organizer region? Because it takes part in nucleolus formation. Okay. Now, in addition to that, there is presence of a rounded knob-like portion beyond secondary constriction, and this rounded knob-like portion is called satellite. Okay. The chromosomes with satellite is called as the SAT chromosomes. It was first discovered by Navaskin in 1912. The satellite was first discovered by Navaskin in 1912. Remember, in case of humans, the satellite is associated with short arm of the acrocentric chromosome number 13, 14, 15, 21 and 22. So just remember that in case of humans, the satellite is associated with the short arm of the acrocentric chromosome number 13, 14, 15, 21 and 22. Okay. So this is what your secondary constriction and satellite. I will discuss about the acrocentric chromosome in the slide or in the video called types of chromosomes. Got it? So now come to the next part, that is the telomere. The telomere is actually terminal end of the chromosome. So end of the chromosome is called the telomere. What is its function? It actually helps in maintaining the integrity of the chromosome. Okay, helps in maintaining the integrity of the chromosomes. In case of most of the eukaryotes, the telomere DNA sequence, that means at the telomere region, the DNA sequence is mostly containing or mostly contains repeats of guanine residues on one strand. So you can understand it by in the following way, that in the chromosome, the terminal end is called as the telomere. So you know that chromosome is made up of DNA. So at the end portion, that portion of the DNA where we say that telomere is found is actually rich in guanine residues. On one strand, if it is guanine residues, then obviously in the other strand there will be what? Cytosine residues. So we can say that it is GC rich region. Got it? Guanine and cytosine region. Now it also provides, your telomere provides stability by preventing the end fusions of chromosomes. So two chromosomes, chromosomal ends do not get fused due to this presence of telomere. So it gives stability to the chromosomes. Got it? Remember it, that shortening of telomere, that means if telomere is a very short part or short one, then it causes senescence and aging. So one of the reasons for aging is the short telomere sequence. Okay, if the telomere sequence is short, then it might be the result of aging. Got it? So you just remember that the chromosomes are made up of what? Your parts called chromatids, then your centromere, which are otherwise called as primary constriction, then your secondary constriction and satellite, then telomeres. So this is a very simple one. So chromatids are the arms of the chromosomes. Each chromatid is made up of a single double-stranded DNA. So after S phase, as the DNA gets doubled or DNA gets replicated, so your chromosome in the metaphasic stage is made up of two chromatids. So at that stage, there are presence of two DNA molecules. Got it? DNA molecule means it's a double. Each DNA is double-stranded. Okay. So in this way, the structure of chromosome the chromosome is ending with telomere, which is rich in guanine residues. The single strand of DNA is containing guanine residues, so obviously the other strand would be having cytosine residues. So this is what the morphology of your chromosomes. Now come to your types of chromosomes. Okay. Types of chromosomes. On the basis of types, you can divide the chromosomes into various types. First, we can say that chromosomes can be of two types, that is autosomes and allosomes. Okay, autosomes and allosomes. Autosomes and allosomes. So what is this autosome? The autosome is actually determining the somatic character or somatic activities of a cell, like growth, development, etc. Somatic means vegetative activities of the cell. Autosomes are concerned with vegetative activities of the cells. So, and as far as allosomes are concerned, they are also called sex chromosomes. Okay, they determine the sexual characters of an organism. In case of human, the allosomes are of two types, X chromosomes and Y chromosomes. So this is one type of division as far as chromosome is concerned. Then we can also divide the chromosomes on the basis of position of centromere. Okay, on the basis of position of centromere, the chromosomes can be of following four types: metacentric, sub-metacentric, then acrocentric and telocentric. Okay, metacentric, sub-metacentric, acrocentric and telocentric. Now come to metacentric. Metacentric chromosome, here the centromere is at the middle position. Okay, and both the arms are of equal length. As you can see in this picture, here the centromere is at the middle position. Centromere is at the middle position. It is the centromere, and the two arms, one arm and two arms, the two arms are of equal length. So you can say that the metacentric chromosome is V-shaped. Okay. Then come to the sub-metacentric chromosome. As far as sub-metacentric chromosome is concerned, here the centromere is slightly away from the middle position. Okay, centromere is slightly away from the middle position and it is L-shaped. Here you can see the picture. This one is the centromere, so one arm is a bit small and the other arm is a bit long. So it is slightly L-shaped. Got it? So this is what your sub-metacentric chromosome. Now come to the acrocentric chromosome. So now the acrocentric chromosome is a somewhat different structure. Here the centromere is sub-terminal in position. That is not really terminal, but a little distance away from the terminal position. So one arm is very long and the other arm is very short. This is what your acrocentric chromosome. As you can see here, the centromere is sub-terminal in position, and one arm is very long and the other arm is very short. So this is what your acrocentric chromosome. Now next one, the last one, is the telocentric chromosome. So here the centromere is at terminal end. So there is presence of only one arm. Okay, and the shape is eye or rod shaped. Okay, so as you can see in this diagram, there is presence of only one arm and the centromere is at the terminal position. So this one is a rod shaped chromosome. Okay, so this is what the types of chromosomes. So we can say that the chromosomes can be of four types on the basis of the position of centromere, that is your metacentric, sub-metacentric, acrocentric and telocentric. So I hope this video would be helpful for you in understanding the simple morphological structure of chromosome. Thank you.